In this last unit, our first lecture is going to be detailing the ways that our kidneys operate. Because the kidneys happen to be the major organs of our urinary system. They're not the only organs, but they are the primary organs. We've also got a pair of ureters, a singular bladder, and then a singular urethra. Function of those organs is basically just either to carry urine, hold urine, or allow for the urine to be carried out of the body. The majority of what happens in the urinary system takes place in our kidneys, and so those will be the things that we talk about the most in this lecture. As you can see here, our kidneys do a lot. And as you guys write down, I'll wait, and then I'll talk about them. Now, some of these things we've talked about before. Okay. For example, the hormones that our kidneys produce, we've talked about both of those before. Calcitriol, we learned, regulates calcium homeostasis of our blood. Okay. Our kidneys release it. And that's going to be a result of the release of parathyroid hormone by our parathyroid hormone when we are hypocalcemic. We've also talked about the production of red blood cells called erythropoiesis, stimulated by the release of erythropoietin from our kidneys. Okay, so that's not brand new to us. Some of the stuff at the top we've talked about, which has to do with the amount, what's in, and the pressure of our blood. Okay? Because that's what our kidneys do. They filter our blood and they control what's in it, which therefore controls the amount of blood we have, which therefore controls our blood pressure. Okay? So when I say composition, I mean what's in it. Okay? So our kidneys regulate our ion composition in our blood. Composition of things like sodium, potassium, calcium chloride, and hydrogen phosphate. Okay. Our kidneys will excrete a variable amount of hydrogen ion and can conserve bicarbonate ions. As we know, bicarbonate ions are very important buffers of pH, okay, by regulating our hydrogen ion concentration. Okay. So that's why we're going to talk about pH balance or acid-base balance in this lecture. Okay. What other two organs help regulate our pH? If you don't know it right now on the top of your head, you better know it come Friday. Your lungs. By controlling how much CO2 we breathe off, right? That's how we regulate pH or acid-base balance in a second-to-second, minute-to-minute, short-term basis. Because we're always breathing from the day we're born to the day we die, right? But we're not always peeing and making urine second-to-second, minute-to-minute. So our kidneys are more important than long-term acid-base balance. And our kidneys are actually the only way that we can excrete out hydrogen ion from our body. So we'll talk about that towards the end of this lecture. Now, when I say volume of blood, what do I mean? Amount. Good. So, if you think back to when we talked about blood, we learned that there were two principal parts of blood. The formed elements, which were about 45%. And then about 55% was plasma. What was the main component of plasma? Water. So we can really affect how much blood we have by simply controlling how much water we pee out. So as it says there, regulation of blood volume is done by conserving or eliminating water in our urine. Now, that also is going to affect our blood pressure. Because if we retain water, what's going to happen to the amount of blood we have? It goes up. And that is going to do what to blood pressure? Go up. Because there's more blood, which means more pressure in our vessels. Okay. The opposite would be if we pee out more water, volume is going to go down, blood pressure will go down. Okay. So it's also very important for that. We talked a little bit about that, but we definitely have talked about the renin-angiotensin-aldosterone mechanism, not in great detail, but we're going to talk about it again in this lecture actually in much greater detail than we have at any other point in the semester. Why? Because your kidneys release renin when our blood pressure is down, because renin and that pathway or mechanism will elevate blood pressure through a variety of mechanisms. The word osmolarity basically is a way to think of our water balance. If you want to think of it real simply, and I listed here as osmolarity. Remember, it's a measure of total number of dissolved particles per liter of solution. Basically, what we're talking about is the balance between water and the stuff dissolved in it. And so that's what we're talking about with regulation of blood osmolarity. Okay. Our kidneys can help regulate our blood sugar levels or our blood glucose levels. Okay. Our kidneys actually aid in that process we talked about the other day, gluconeogenesis, which, as we learned the other day, is the formation of glucose from non-carbohydrate sources. In this example, our kidneys use the amino acid glutamine to make glucose. Our kidneys can also release glucose through our urine from our blood to help maintain blood sugar levels, but you should not have a lot of glucose in your blood, or in your urine. Okay. One of the tests they do to determine if somebody is hyperglycemic or possibly diabetic is measure glucose in their urine. And because that's very common for people to excrete a lot of urine or a lot of glucose in their urine if they're diabetic, because their kidneys or their pancreas, as we know, is either not producing insulin or not. Their body's not using it. It's becoming insulin resistant. And then excretion of waste, which we're going to talk more about waste here on the next page, are formed from metabolic reactions and can be foreign substances like drugs or toxins that we must get out of our body. It includes things like ammonia, urea, bilirubin, creatinine, uric acid, various drugs, and environmental toxins. Let's talk more about these wastes. We've talked quite a bit actually about waste fairly recently with the digestive system. We talked about those wastes. And even carbon dioxide is a waste. A waste is basically just any substance useless to the body or it's present in excess of the body's needs. Now, a lot of wastes are a result of metabolism, like carbon dioxide is an example of a metabolic waste, produced during the Krebs cycle and the transition reaction, okay, during any form of metabolism or chemical reaction. Okay. Nitrogenous wastes are examples of metabolic wastes that contain nitrogen, and we've got several of them over here on the right. The other day we talked about ammonia and urea, actually. Urea, which is formed from the catabolism of proteins. Remember the other day we talked about deamination, where we take an amino acid and we break it apart, removing the amino group. We talked about how we form ammonia as a result of that process, but that our liver will convert that ammonia to urea. And ammonia is very toxic, urea is less toxic. But we still don't want urea in our blood, so our kidneys are going to filter it out and make it into urine, or put it into our urine. Same thing for uric acid and creatinine. Uric acid, byproduct of nucleic acid metabolism. So the breakdown of DNA and RNA. You've heard of gout before? Gout happens when you're not getting rid of your uric acid. And then creatinine, we've talked about that before, comes from the breakdown of creatine phosphate, which is produced in our skeletal muscle and is used as an energy substrate. So that's what happens when we take care of our waste. Now, normal, healthy individuals not going to have any problems getting rid of their waste. What can happen, though, if somebody has renal insufficiency or kidney failure, is a condition called azotemia. Azotemia is defined as an abnormally elevated BUN. BUN is short for blood urea nitrogen, a measure of the amount of or the level of nitrogenous waste in our blood. It basically is an indicator if the kidneys are working correctly. The word renal, as you guys hopefully know by now, is a term that is interchangeable with kidneys. And if the kidneys are not functioning to their maximum or not functioning nearly enough, it may require a process called hemodialysis or dialysis to be performed. Okay, and I'm sure you guys have all heard of dialysis before. This is a process that people that have kidney failure or some form of kidney disease need to have done on a regular basis, okay, every couple days. Otherwise, they're going to be very, very in danger. What happens is they are hooked up to a machine with two lines, and there are various, in this case, arm, could be somewhere else in the body. But one line draws blood out into the machine, that then runs it through and filters it out. We can kind of call it the dirty blood if you want to. And then the dialysis machine will clean it, give it new solution. The plasma is basically cleaned. And so the clean blood, as we'll call it, is then shunted back into the body through the other line. So basically, this just replaces what our kidneys do. And speaking of our kidneys, the structures that do the filtering and cleaning of our blood are called nephrons. So you guys have all taken anatomy. Hopefully you guys remember from last semester when we talked about those things called nephrons. Each of our kidneys has over 1 million of them. They're very small microscopic structures made up of two principal parts. Okay, we're not going to get to talk about what they do completely, but they perform the three major things that eventually lead to the formation of urine. Okay, the first process takes place within the renal corpuscle. Okay, the renal corpuscle is shown right here. This is a very simplistic version of a nephron, by the way. We'll show you a more detailed one here in a second. But within a corpuscle is where we first filter out stuff from our blood, what we call filtrate, which then drains into this long winding tube, what we call the renal tubule. Okay. Renal tubule consists of three major parts: a proximal convoluted tubule, or it's just listed here, proximal tubule, the loop of Henle, and then a distal convoluted tubule, or just distal tubule. Okay, that's where we then perform the other two functions, or processes of urine formation. Okay, we finalize the formation of urine in our collecting ducts, which a collecting duct is kind of part of a nephron, but it's kind of not. See these little branches here? Those would actually be the distal convoluted tubule of another nephron. So collecting ducts are actually shared between several nephrons. Okay, and by the time we reach the end of a collecting duct, it's urine. Okay, it's no longer called filtrate. Okay. What we're going to do here on the next 2 pages is zoom in and talk about, here on the very next page, the corpuscle, and then we'll talk about the tubule on the following page. So here is a zoom in at that corpuscle, much more detailed than what we saw on that previous page. This is where blood is first brought into the nephron by way of what's called an afferent arteriole. Okay. These afferent arterioles are very long, distant. Let's just say this. They're branches of the renal arteries. Okay, your renal arteries are branches that come off of your aorta. You have a right and a left that feed your right and left kidneys, respectively. They go through some forking and branching. Okay. The ones that actually run into our nephrons are called afferent arterioles. Okay. And they come into the capsule, what we call the Bowman's capsule, which is what surrounds our corpuscle. Okay. And that renal or that afferent arteriole forks or branches into this network, what we call a glomerulus, or sometimes we'll call them glomerular capillaries. Okay. But you can't really see those in this picture. There's a little red that you can see in there, but you can see they're covered by these yellow things that look like little bugs. Okay. Those things are called podocytes, which wrap around the capillaries. Okay. Now, I call them little bugs because they look like little bugs, and like little bugs, they have little feet-like extensions, or little legs. Those legs are called pedicels. Okay? And I like to think of them like what I'm doing with my fingers right now. where I've got them kind of intertwined with one another. If everybody wants to look up from their phones and look at me. Okay. Your fingers, my fingers, I should say, if I were to have somebody or go over to the sink and pour water into my fingers, would not completely hold the water, would they? Okay. They're going to allow water to flow through. Okay. So they're not watertight. But if I were to walk outside and find some big sticks and rocks and have somebody dump them into my hands, there's a good chance that they're going to stay in my hands and not fall through. So they're going to allow small things through, but not big things. Well, what's small? Water, ions, wastes, glucose. What's not small are your cells. So red blood cells, your platelets, your white blood cells, and then your large plasma proteins. Okay. So we basically pull out the plasma from the blood, and it collects into this space here, what we call the Bowman space, or just the intracapsular space. And then that filtrate flows into the beginning of our renal tubule. Okay. What's left in our blood exits our renal corpuscle as an efferent arteriole. And at that point, it's basically a lot less water, and mainly it's going to be the blood cells. What we're going to find out is that water and a lot of that stuff we filtered out here actually will get pulled right back into our bloodstream in this part of our tubule, what we call the proximal convoluted tubule. And that's the very beginning of our renal tubule. This renal tubule is a winding hollow tube extending from the renal corpuscle to the end of the nephron, where it joins the collecting duct. And like I said, the collecting duct is shared between many other nearby nephrons. We can describe our renal tubule as the urine collecting and concentrating tube. The first part is called the proximal convoluted tubule. Anybody know what the word convoluted means? It's called a jumbled up mess, basically. And that's what convoluted means. Here you can see what I'm talking about. Here's our corpuscle. We don't see the podocytes, so we can actually see the capillaries, the glomerulus here. Here's our afferent arteriole where blood's brought in. We filter it out here in the corpuscle, and then it filters out into or drains out into our proximal convoluted tubule. Now the efferent arteriole, which is the skinnier of the two between the afferent and the efferent, will exit the corpuscle, and then it forks to become these what we call peritubular capillaries, which, as you can see, are the little red strings that are surrounding the entirety of our tubule. This is where stuff can get brought right back into the bloodstream through a process called reabsorption, or where we can pull stuff from the blood into the tubule. Okay, I mentioned we pull out most of the water and ions here. We actually end up putting them right back in, right in this part of our tubule. And like 99% of that water that we filtered out gets put right back in. And you'll see why. Now, the cells that are found in the proximal convoluted tubule are simple cuboidal with numerous mitochondria and a prominent microvillus. We can actually see that right here on the previous page. Here's our simple layer, so singular layer of cube-shaped cells. We talked about microvilli the other day when we talked about digestion, were those little finger-like extensions. We talked about those because we find those in those absorptive cells of our small intestine, which provided a lot of ways to get stuff into the intestine from the lumen and a lot of ways to secrete and increases the surface area. So what actually happens is that filtrate leaves and enters our proximal convoluted tubule. This figure doesn't show it, but there are those peritubular capillaries directly outside of the tubule. And these cells basically pull that water right back into the blood, along with a lot of those ions. So it kind of seems stupid that we filter and then put it right back. That's what we do. Okay. Our proximal convoluted tubule is contained only in what we call the renal cortex. The renal cortex is the very outer part of our kidneys. Okay. Now our second segment of the renal tubule dips down into the medulla, which is the middle part of our kidneys, and we call this segment our nephron loop or loop of Henle, which includes a descending limb that goes down, carrying that filtrate downwards, and then an ascending limb that carries it upwards. Okay. If you look hard enough, you'll see that part of our loop of Henle is really thin. The tubule here that I got my little arrow on is very thin. The thin segments are very permeable to water, which means water will pass through them really easily. Okay. They're thin because the cells here are simple squamous, which means that the cells are a single layer thick and they're flat plate-like. Okay. Whereas the thicker segments, which you can see right over here in our ascending limb, it's all real thick. Okay. The cells there are cuboidal, but they're bigger and thicker because the cells have a lot of mitochondria in order to produce ATP. And because they're undergoing what's called active transport. And we know what is needed for active transport? ATP. Because we're pumping against the gradient. What we do here is actually pump out salt, which is going to draw water out from our tubule over here. So we're going to talk about what's called the countercurrent multiplier mechanism. Okay, it happens in our loop of Henle. From there, the filtrate travels back out into the cortex and meets another kind of jumbled up tube, what we call the DCT, distal convoluted tubule. Okay. Now, as we pass that filtrate through the distal convoluted tubule, look what it happens to pass right by that afferent arteriole. Okay. If I go back to the picture of our renal corpuscle, here we can actually see the distal convoluted tubule. We're only showing a very small segment of it. But look how close it is to the afferent arteriole. This is where the blood first came in to form filtrate. Okay. There's a relationship between these cells, what we call macula densa cells, and these cells, what we call juxtaglomerular cells, where they can communicate with one another to make sure the production of urine is going correctly. Okay. I have an analogy. It seems to work pretty well. Okay. Think of the process of urine formation like a conveyor belt, and we're making Barbie dolls, okay, where each station pops on a different limb or a head to the Barbie doll. You know, somebody's putting the clothes on, somebody's adding accessories, all that wonderful stuff. Okay, and let's say the conveyor belt gets sped up to a point where somebody down the line is not keeping up. He's got a little switch at his station that he can push to slow down the entire conveyor belt. So it feeds back all the way to the beginning of the conveyor belt. That's basically what these cells can do, as can these cells. If we're making our belt too fast, the filtrate's being formed too quick, and we're passing the urine too quickly through, we're going to end up losing water or salts or whatever through our urine. We don't need that to happen. So by passing by what we call the juxtaglomerular apparatus can help maintain the formation of filtrate and therefore the formation of urine. So let's read through the juxtaglomerular apparatus. Portion of the distal convoluted tubule brushing past the afferent arteriole. Interaction will help maintain the homeostasis of blood flow. We have granular juxtaglomerular cells, which are specialized smooth muscle cells of the afferent arteriole, and they contain mechanoreceptors monitoring blood pressure in the afferent arteriole. The more important of the cells are the macula densa, because they are the cells of the distal convoluted tubule, and they contain chemoreceptors monitoring the concentration of sodium and chloride ions in our urine inside the tubule. So they can control how much salt, basically, we lose. And that also influences how much water we lose. Because as we'll see in this lecture, water and salt kind of go hand in hand. Okay. Now, from our distal convoluted tubule, urine, at this point it's not quite urine, but it's pretty close. We'll still call it filtrate, drains into a collecting duct. Now we have special cells in the collecting duct and the very end of the distal convoluted tubule that finalize the formation of urine. Principal cells, they're going to respond to two hormones, aldosterone and antidiuretic hormone. Okay. Anybody remember what the nickname for aldosterone was? It was our blank retaining hormone. Good. It retains salt. So that means it's going to tell our collecting duct in the very end of our distal convoluted tubule to pull salt out of the tubule and put it back into the bloodstream so we don't pee it out. What does antidiuretic hormone make you do? Retain water. Same thing, except for instead of salt being retained, we're pulling back in water. And that's thanks to the principal cells. The other important cells are called intercalated cells, and they're going to help us secrete hydrogen ion, and that's going to be lost through our urine to help us maintain long-term acid-base balance. Okay. We're going to go through these. I ran you through a real quick explanation of urine formation, but here is a more detailed explanation of those three steps. And this involves both the nephrons and collecting ducts. Begins with the formation of glomerular filtrate. So that's why they call it glomerular filtration. This is where water and most of our solutes in the blood move across the wall of our capillaries into that capsular space and then drain into the renal tubule. Okay. What doesn't leave the blood includes the blood cells and proteins, which exit to the efferent arteriole, which then becomes the peritubular capillaries. Okay. So if you haven't paid attention or haven't figured it out yet, this is a very simplistic drawing of our nephrons. Okay. In our tubule, we do two things. We pull stuff from the tubule back into the blood, which is what we call reabsorption. Okay. We've talked a lot about absorption lately. We did that in our small intestine, our stomach, and our large intestine. Small intestine is where we did most of our absorption. Okay. Like if I were to take a drink of my water, eventually that water is going to get pulled from the lumen of my tract into the bloodstream. That first point when water is brought into the body is called absorption. And that's what our digestive system is responsible for. But our kidneys allow for reabsorption, which is where that water that was lost here can get pulled back into the blood. Remember, and this is common knowledge, if you throw the letters R-E at the beginning of a word, like redo, it means you're doing something a second time. So we're reabsorbing means that we're pulling stuff back into the blood, maybe a second, third, fourth, fifth time. Okay. And we've talked about reabsorption. Okay. Back when we learned about blood vessels, we mentioned reabsorption was the return of substances to our bloodstream. Okay, so that's not brand new to us. Secretion is another term that we learned about previously. Okay. And I like the fact that they keep the arrow here, or the color of the arrow here, yellow. Because if you think about it, stuff going into the tubule is eventually going to become urine. And what color is urine a lot of times? Yellow. And so secretion is where stuff leaves the blood and enters our tubule. And we do a lot of back and forth, a lot of reabsorption and then secretion. All the way from the beginning of that tubule all the way to the collecting duct. And then from the collecting duct, it's urine that drains into a series of tubes all the way down into our ureters, which will then travel it or carry it to our bladder. Okay. Let me read you the last paragraph here. Solutes in the fluid formed by nephrons and collecting ducts drain into ureters as urine to be excreted out of the body. The rate of urinary excretion of any solute is equal to its rate of glomerular filtration. plus its rate of secretion minus its rate of reabsorption. So it's how much of this minus this plus this. By filtering, reabsorbing, and secreting, our nephrons will help maintain homeostasis of our blood's volume, which therefore also affects composition. Okay. Now, we're not going to get all the way through, but I want to talk a little bit more about each of these steps in greater detail. So we'll first talk about filtration, which once again is a process in which water and some solutes in our blood plasma pass from our capillaries of the glomerulus across a 3-layer filtration membrane into the capsular space of our renal corpuscle. The fluid formed here is called glomerular filtrate. Okay. What we have pictured down here is a small segment of our glomerular capillaries and the podocytes surrounding them. So we basically just took a little chunk out of our net there. Okay. If we look, we see that the capillary itself, its wall is very, very porous. Okay. It has a lot of fenestrations or pores. Okay. These large filtration pores are very highly, or make the capillary highly permeable to many substances. What it will not be permeable for, or to, which means permeable, once again, stuff to pass through it, white blood cells, red blood cells, platelets, and proteins. The presence of red blood cells, proteins, white blood cells, etc. in our urine indicates either that there's a problem in pressure, because this is related to pressure, okay, or it could mean that there's a defect in the filtration membrane. Okay. To help these glomerular capillaries, we have what are called mesanglial cells. They're found between the capillaries as well as in the cleft between the afferent and efferent arterioles, and they can contract to help regulate this filtration process. Okay. If I go back to that picture, here you can see the mesanglial cells. You can also see them just a little bit in here. Okay. We'll talk more about their role with pressure next week when we talk about this more. Okay, directly surrounding that layer is a basement membrane, or basal lamina, which is acellular, so it's a non-cellular material, made up of minute collagen fibers and proteoglycans in a glycoprotein matrix. A lot of big words in there, not that important. What is important is this last sentence. They have a negative charge, and what do we know about things that are also negative charged? Or like charges. They are not attracted to one another, are they? Because opposites attract. Likes repel. A lot of our plasma proteins are negatively charged. So even if they can fit through these little holes, they're not going to want to, because of these, or this membrane that is also negatively charged. So it's going to repel them. Okay. And then the outer layer is made up of those podocytes with those little slits. Okay, but we have a membrane that you can see now that we've zoomed in called a filtration slit that spans across. So it's not quite like my finger analogy, but it would be like if I had like a coffee filter between and the gaps between my fingers. Okay. Now, this filtration slit and the membrane there is also going to carry a negative charge, also preventing any large negatively charged objects from passing through. Okay. What can pass through this very thin membrane are things with a diameter smaller than 0.006 to 0.007 microns. That's so small, it's unbelievable. Okay. What is going to qualify there? Water, glucose, vitamins, amino acids, your smallest plasma proteins, ammonia, urea, and ions. Okay. For most things, they're going to get stuck in our blood. But if you think about it, water, solutes, They make up the bulk of our plasma, with the exception of our plasma proteins. And if our plasma proteins in our blood cells don't pass through, you can basically think of the stuff that leaves our glomerulus as plasma proteins in red blood cells, and that's about it. White blood cells, platelets, that's about it. So the filtrate, in other words, if you want to think of it this way, basically is plasma without any plasma proteins. So we'll get into this in greater detail. And like I said, it's related to pressure. So we'll talk more about pressures in the relationship of glomerular filtration on Monday, since we are at it.